A team at UC Berkeley ran the numbers on a completely different data set. Not ancient bones, not cave sediment, living people. 2,762 of them, drawn from across the most genomically complex population on the planet. And what Dr. Priya Morjani and her colleagues found stopped the field cold. India. The answer was India. Not a cave in Croatia, not a frozen specimen from Siberia. A retired schoolteacher in Lucknow, a farmer in Tamil Nadu, a woman in Andhra Pradesh whose grandmother still knew the name of their village from 4 generations back. By assembling the fragments of Neanderthal ancestry distributed across 2,762 living Indian genomes, Morjani's team recovered approximately 1.5 gigabases. That's roughly 50% of the entire introgressing Neanderthal genome, more than any study in history had ever managed. Published in Cell in 2025. If you want to go deeper into this entire story, the full research, the science behind it, and what it means for your own ancestry, I've put together a detailed ebook covering everything. The link is in the description. Go grab it. Now, let's get into the story. This isn't just ancient history. This is in someone's blood right now. And here's the question that doesn't go away. What happened to the Indian genome that made it capable of this? What combination of time, migration, mixing, and isolation produced a genetic archive so layered, so preserved, so complex, that it holds the answer to a question about an extinct species that died 40,000 years ago? If you've ever genuinely wondered where humans actually come from, not the simplified version, but the real biological story, subscribe now, because what we're about to go through is not the version most channels are willing to tell. The starting point is a grave. Rakhigarhi, Haryana, India. Somewhere between 2,800 and 2,300 BCE, a woman was buried among dozens of ceramic bowls and vases in the dust, northwest of what is now Delhi. When Dr. Niraj Rai of the Birbal Sahni Institute of Paleosciences in Lucknow finally extracted viable ancient DNA from her remains, and he will tell you it was the most intensive effort his team had ever made on a single sample, her genome showed something specific. Her mitochondrial haplogroup was U2B2. Her ancestry was a mixture of ancient Iranian-related lineages and a component from South Asian hunter-gatherers. And her steppe ancestry was exactly 0, not a trace. She lived at the height of the Indus Valley Civilization, one of the first urban societies on Earth, and she carried none of the genetic signal that would arrive from the north, later. The fact that we know this at all is one thing. What it means is something else entirely. 50,000 years ago, a group of humans made a turn, not a metaphorical one, a geographic one, up into the subcontinent. And the question that takes 6 more segments to answer properly is this. When they made that turn, did they find anyone already there? Already there. That phrase carries more weight than it looks. The Nalamala Hills, Andhra Pradesh. Right now, not in the ancient past, right now, there is a semi-nomadic community called the Chenchu living in the same forest ranges their ancestors used for tens of thousands of years. They hunt, they gather. They have largely resisted the kind of cultural integration that erases older ways of life. And when geneticists sequenced their genome, they found something that no one had a clean explanation for. Between 70 and 80% of their ancestry traces to a population that doesn't exist anymore. A population with no surviving unmixed descendants anywhere on the mainland of India. Scientists call it AASI, Ancient Ancestral South Indians. A genetic lineage that formed approximately 40,000 years ago on the Indian subcontinent and then stayed there, isolated from everything that was happening in Europe, the Middle East, Central Asia, all of it, for tens of thousands of years. But here's where it gets strange. When researchers looked for the closest living analog to AASI, some population today that could serve as a reasonable genetic proxy, the best they could find were the Andaman Islanders, the Anj, people who have lived on islands in the Bay of Bengal for so long, in such isolation, that even they have drifted far from the original AASI signal. Far enough that geneticists classify them as an imprecise substitute. The AASI lineage doesn't really have a living equivalent anywhere on Earth. It is uniquely South Asian, and it is a ghost. This is the work of Dr. Vageesh Narasimhan at UT Austin and Dr. Nick Patterson at Harvard, working alongside Priya Morjani and David Reich's team. In 2019, they sequenced 523 ancient humans from across South and Central Asia. And what they traced in that data was a genetic gradient, a spectrum running from ancient Iranian hunter-gatherers on one end to a lineage related to Southeast Asian populations on the other. And at the South Asian end of that spectrum sat AASI, distinct, isolated, old in a way that makes the Indus Valley Civilization look recent. The Arula people of Tamil Nadu carry more than 70% of this lineage in their living genome. They are the closest mainland window we have into who was walking the subcontinent when Europe was still under ice sheets. And that window comes with a specific address, haplogroup HM69, a paternal Y chromosome lineage found at 15 to 30% frequency in Dravidian-speaking tribal populations like the Chenchu, the Gond, and the Arula, found nowhere else on Earth at meaningful frequency. It is the molecular fingerprint of 65,000 years of South Asian presence. Tens of millions of living people carry it. Most of them don't know its name. Between 5,400 and 3,700 BCE, something happened in the borderlands between Iran and South Asia, a mixing event of enormous consequence. A people formed who would go on to build the largest civilization of the ancient world. They left no name for themselves. Their descendants carry the most genomically complex ancestry on the planet. And in 2019, scientists finally held their DNA in a centrifuge tube the size of a thumb. Their DNA arrived in a laboratory in 2019 inside a centrifuge tube the size of a thumb, and what came out changed the field. Rakhigarhi, Haryana, 2019. Dr. Vasant Shinde of Deccan College in Pune has been excavating this site for years. He is standing over Trench 7 when his team uncovers her. The skeleton, the ceramic bowls arranged around her, the grave goods that place her unmistakably inside the Indus Valley Civilization at its height. Somewhere between 2,800 and 2,300 BCE. Adult. Probably female based on her DNA. Getting viable ancient DNA out of South Asian burial sites is, to put it plainly, brutally difficult. The heat, the volatile soil chemistry. Most samples degrade beyond any usable signal. This one held. And when Dr. Niraj Rai's laboratory in Lucknow finally completed the sequencing, the genome showed something that forced a complete rethink of where the Indus people came from. She carried 45 to 82% ancestry related to ancient Iranian farmers. She carried 11 to 50% of that older AASI hunter-gatherer component. And she carried 0 steppe pastoralist ancestry, 0 Anatolian farmer ancestry, no genetic signal from the West, at all. And yet, the data said the exact opposite of what the standard migration model predicted, because the Iranian-related ancestry in her genome wasn't from Iranian farmers the way we usually think about them. The lineage in the Rakhigarhi women had split from Iranian populations before farming was invented in the Fertile Crescent. Before. Which means the connection between the Indian subcontinent and the Iranian plateau is older than agriculture itself. These weren't farmers who arrived and brought their crops. They were people who mixed with South Asian hunter-gatherers before anyone in that region had learned to plant a seed. Farming in South Asia may have been invented independently, or it arrived later as an idea, not as a wave of people. This is not a minor revision to the textbook. This rewrites the chapter. What the Indus Valley Civilization represents genetically is a consolidation, not a beginning. By the time Mohenjo-Daro was a functioning city with standardized drainage and 10,000-person neighborhoods, the genetic foundation of its population had already been forming for thousands of years. They built in standardized bricks, ratio 4 to 2 to 1. Found identically proportioned at Mohenjo-Daro, at Harappa, at over 1,500 other sites across a territory the size of Western Europe. The people who made that brick are in the Rakhigarhi genome. Their descendants are in the Morjani 2025 study. The brick survived. The name did not. The Indus script, the writing system they left behind, has never been deciphered. We know what their city smelled like after rain. We know the dimensions of their bathing platforms. We cannot tell you what language they spoke. We cannot tell you what they called their gods or themselves. The genome is the only language of theirs that survived intact, and it contains one more thing, something that came from the north, something that changed the paternal record permanently. The men who built Mohenjo-Daro, if their Y chromosomes could speak, most of them have gone quiet. What replaced them? That answer is 4,000 years old, and it is riding towards South Asia on horseback from the Eurasian steppe. From the north, 4,000 years ago, on horseback. The Yamnaya pastoralists, and if you've been following this channel, you know this name, had already swept west across Europe. Their genome, defined by haplogroup R1a, had already reshaped the paternal landscape of the British Isles, Scandinavia, and the Pontic steppe. And then the expansion turned south. Through the Bronze Age Sintashta culture of modern Kazakhstan, through the Hindu Kush, down into the valleys of what is now Pakistan's Swat region. What Narasimhan and Patterson's 2019 team found in those Swat Valley burials should have been major news. Among the late Bronze Age and Iron Age individuals they recovered, people who lived between 1500 and 500 BCE, 44 Y chromosomes were analyzed. Only 5% of them showed the steppe-associated R1a-Z93Y chromosome type. 5%. But when those same individuals' autosomal DNA, the non-sex chromosomes, was analyzed for steppe ancestry, the number was 20%. 4 times higher on the autosomes than on the Y chromosomes. That is not a rounding error. That is a z-score of minus 3.9 standard deviations. In genomic statistics, that means you have detected something real with near certainty. What it detected was this. In the early wave of steppe contact in South Asia, the ancestry entered these communities predominantly through women, not conquest, not a wave of male warriors displacing local men. The first signal of steppe ancestry in South Asian bodies came through female movement, steppe women, or their daughters, being incorporated into IVC successor communities through channels that the genome cannot name. Marriage, trade networks, we don't know. Only the asymmetry survives. Stay with me, because this next part is the reason this video exists. Because the present-day picture inverts. In modern South Asians today, the pattern goes the other direction. There is a statistically significant excess of steppe-related Y chromosome ancestry compared to autosomes. Z equals positive 2.7, which means the paternal lineage replacement came in a second, later wave. Steppe men arriving and leaving their Y chromosomes in much higher proportion than the rest of their genome. 2 waves, 2 opposite sex biases. The first, steppe women entering IVC communities. The second, steppe men replacing the paternal record of those same communities. Haplogroup R1a-Z93 is now found in approximately 17.5% of South Asian men, dominant among Indo-European speaking populations in northern India. The men who carried this marker 4,000 years ago rode across Kazakhstan and had never seen the Indian Ocean. Their Y chromosome is now inside the bodies of tens of millions of men who were born in Mumbai and Chennai and Lahore, who assume their ancestry is straightforwardly local. I want to be careful here because this data sits at the center of a debate that has been deeply politicized for over a century. The genome does not take political positions on what happened. It doesn't use the word Aryan. It doesn't have an opinion about the Vedas. It keeps a different kind of record. And what the record shows is not a simple replacement. It is 2 asymmetric waves that together produce the population structure that everyone from ancient historians to modern geneticists has been trying to explain. Actually, hold on. Before we go any further, I want to know something. In our last video, we followed the Yamnaya expansion west across Europe. And now we're watching the same genetic wave arrive in South Asia, except the sex bias data here completely changes what migration even means. Did the steppe expansion feel different to you heading east versus heading west? Drop that in the comments, because the asymmetry changes the story for the next 3 segments. For 150 years, scholars debated whether the composers of the Rig Veda were the same people who built the Indus Valley Civilization, or a separate incoming population that replaced them? The genome has an answer, and it contradicts both sides of that argument in a way that neither side is going to find comfortable. Both sides were wrong, but not in the way either expected. The Swat Valley, Iron Age. The burials here show hybrid paternal lines, some local, some steppe. But when you look at the autosomal picture, the people themselves are not replaced. They are combined. The incoming steppe ancestry didn't erase the IVC foundation. It layered over it. And when the IVC declined, between roughly 4,000 and 3,500 years ago, its people didn't vanish. They split. One portion moved southeast, mixing with populations already living in peninsular India, to form what geneticists call the ancestral South Indians, AASI. The other portion mixed with the incoming steppe pastoralists to form the ancestral North Indians, ANI. And here's the number that stops the argument cold. ANI ancestry today ranges from 39 to 71% across Indian populations. ASI makes up the remainder. Between them, they account for virtually everyone. But both of them, ANI and ASI, owe their primary ancestry to IVC-related people. Everything I just told you? Hold on to it, because it's about to mean something completely different. Because what this means is that the people who built Mohenjo-Daro are the primary ancestors of the people who wrote the Vedas, and the primary ancestors of the people of peninsular South India, whose civilizations are typically described as entirely separate from the Vedic tradition. Same source population, two different mixing partners, two apparently different cultural worlds, and at the genomic bedrock, a family argument, the Aryan-Dravidian divide. The distinction that has fueled linguistic nationalism, caste ideology, and political identity across South Asia for over a century. The data from Narasimhan, Patterson, Moorjani, and Reich doesn't say it's trivial. It says it is downstream, a branching of what was, at its roots, one population responding to two different mixing events in two different directions. Now, here's the part nobody talks about. David Reich's team, working with Nathan Nakatsuka and colleagues at Harvard, ran a different kind of analysis on Indian populations. They measured founder events, the genomic signature left behind when a population sharply reduces in size and then maintains strict endogamy, meaning marriage exclusively within the group. They sampled 263 South Asian population groups, and 81 of them showed founder events as strong as, or stronger, than the Finnish and Ashkenazi Jewish bottlenecks. Those two populations are the most studied genetic bottlenecks in European medicine. 1/3 of the groups surveyed in India matched or exceeded them. 26 groups were more severe than the Ashkenazi. Collectively, tens of millions of people. One of the most striking, the Visya, a middle caste community in Andhra Pradesh, approximately 5 million people. Their bottleneck, the moment their ancestors sealed themselves into a closed genetic community, dated to between 3,000 and 2,000 years ago. After that point, average gene flow from outside their group fell below 1 in 30 per generation. For 2,000 to 3,000 years, 5 million people living within touching distance of other Indian communities, and genetically as isolated as if they were on an island. The IVC people were not replaced. The steppe migrants did not erase them. The merger produced two streams of descendants, and then something came from within those descendants that changed everything again. Not an invasion, not a collapse, a lock from within. In the Swat Valley, we can see exactly when, from within, begins to look like something else entirely. Her burial catalog number is SWA-001. She lived in the Swat Valley in what is now Pakistan, sometime around 1300 BCE, deep in the period when steppe ancestry was actively integrating into South Asian communities. The archaeologist who excavated her grave noted the isotopic evidence in her bones: a varied diet, grain, meat, some marine resource, an ordinary life in the sense that most lives are ordinary to the person living them. She was not ordinary to the genome database. Her autosomal DNA carried approximately 20% steppe ancestry, a meaningful fraction, but her mitochondrial haplogroup, the direct maternal lineage, mother to mother to mother going back through time, was entirely local, pre-steppe South Asian, unbroken. Which means the steppe ancestry in her autosomal genome almost certainly did not come through her mother's line. It came through her father, whose own mother was likely a steppe migrant or descended from one. 3 generations, a family, not an event. Narasimhan's team, analyzing the sex bias statistics across the Swat Valley sample, found the asymmetry at z equals -3.9, the signal of steppe ancestry entering these communities predominantly through women in the early wave. SWA001 is the statistical average made human. She is the proof in one skeleton that the first steppe ancestry to reach South Asia did not arrive on horseback with a spear. It arrived through women whose names were never recorded, whose migrations were never documented, and whose contribution to the genome of 1.4 billion people was invisible to every historical source that came after them. This discovery was made in 2019. Almost nobody talked about it, until now. The entire debate about whether the steppe migration was an invasion, or a diffusion, or a cultural exchange, every argument in that debate uses male-gendered language. Male warriors, male priests, male chieftains spreading Indo-European languages. SWA001's genome argues that the early mechanism was something else. The historical record, written by and about men, missed the wave that arrived first. Her mitochondrial line connects her directly to the population that was already here. Her autosomal steppe ancestry came through a father whose biological story crosses the Hindu Kush. She did not experience her life as a data point at the intersection of 2 civilizations. She experienced it as a family. 3 generations of people making specific choices that we cannot reconstruct, and leaving, in her bones, a record that lasts 3,300 years. The genetic boundary between the ANI cluster, Northern Indian ancestry, and the ASI cluster, Southern Indian ancestry, maps directly onto a geographic and social fault line that is still structurally active in 21st century India. The genome didn't create that fault line. But in the bones of the Swat Valley, in the mathematics of that z-score, we can see exactly when the fault line was poured, hardened, set, and never renegotiated. Poured, sealed, hardened. The endogamy lock closes between 3,500 and 2,000 years ago, and when it does not close on outsiders. It closes on neighbors. Andhra Pradesh, the Visya community. Approximately 2,500 years ago, roughly the time of the early Iron Age in South Asia, something happens that leaves no monument, no inscription, no chronicle. A group of several hundred or a few thousand individuals begin marrying exclusively within their group. The decision is not dramatic. It is probably practical, social, a way of preserving resources, status, identity. It is the kind of decision that feels local and specific and rational in the moment it is made. David Reich's team looked at the genomic homozygosity, the runs of identical DNA segments, in the Visya today. They called the result shocking. After the bottleneck, gene flow into the Visya community fell below 1 in 30 per generation, and it stayed there for 2,000 to 3,000 years. They were not living in isolation geographically. They were living inside one of the most densely populated regions on Earth, surrounded by other communities, trading, traveling, interacting, and genetically sealed. There are 3 things this study revealed. The third one is the one that changes everything. The first, 81 out of 263 sampled South Asian population groups show founder events as strong as or stronger than the Finnish and Ashkenazi bottlenecks. The second, 26 of those groups are more severe than the Ashkenazi event, collectively representing tens of millions of living people. The third, the same genetic isolation that preserved every layer of South Asian ancestry, the AASI component, the Iranian farmer layer, the steppe signal, is also the mechanism that allowed that Neanderthal reconstruction. Because the endogamy lock didn't just preserve ancestry, it preserved variation. Different communities sealed different combinations. And when Moorjani's team assembled those combinations across 2,762 genomes, They had 50% of the Neanderthal genome. Now hear this clearly. The Harappans did not vanish. The Harappans sealed themselves. Between 3,500 and 2,000 years ago, the inheritors of the world's first urban civilization, the people whose biological foundation runs through both the ANI and ASI streams, locked themselves into genetic compartments so rigid that today, some of their descendants are as genetically different from each other as Europeans are from East Asians. Not because of oceans, not because of mountain ranges, because of a social contract written before iron was smelted in India. And that contract is still running, in living cells, right now. The genome cannot tell us who wrote that contract. It cannot tell us whether it was imposed or chosen. It cannot tell us whether the people who lived inside it experienced it as protection or as constraint. What it can tell us, with a z-score and a runs of homozygosity analysis in 2,762 sequence genomes, is that the decision made on an ordinary day in Andhra Pradesh 2,500 years ago has a living medical consequence that the global healthcare system is only now beginning to see. In at least one community, the genomic clock places the endogamy bottleneck 800 years earlier than the earliest written historical reference to that group's caste identity. The lock engaged before the name for it existed. Caste. The word the genome cannot say. The genome only speaks in frequencies and distances. A genetics clinic in Chennai, Tamil Nadu. A young woman's screening has returned a flag, a rare recessive variant with elevated frequency specifically in her South Indian community. Her clinician opens the standard reference databases. The variant is not there. It was first documented in 2024, and it has been sitting in that community at that elevated frequency since the founder event that sealed her ancestors into a closed genetic group approximately 2,000 years ago. This is not an edge case. When Kurdankov, Skov, and Morjani's team finished processing their 2,762 Indian genomes in 2025, they identified 24 million novel single nucleotide variants, genetic markers not present in any existing global human reference database. Not the 1,000 Genomes Project, not gnomAD, the most comprehensive variant database in existence. 24 million novel variants, over 99% of them rare. But rare in a population of 1.4 billion means tens of millions of people carrying genetic information that the medical system has never cataloged, cannot interpret, and therefore cannot use. This isn't just ancient history. This is in your DNA right now. If any part of your ancestry traces to the Indian subcontinent, any fraction, any generation, some portion of your genome is inside that 24 million variant library. It is not in any database your doctor can access. The precision medicine revolution, pharmacogenomics, disease risk prediction, personalized dosing, was built primarily on European and, to a lesser extent, East Asian genomes. The South Asian genome is the exception in the equation that the equation was not built to solve. Think about what that actually means. The same endogamy lock that preserved the AASI ancestry, the oldest human layer in South Asia, 65,000 years undiluted, also concentrated deleterious recessive variants in specific communities. Conditions that sit below the clinical radar, because the communities carrying them were never represented in the global genomic studies that set the radar's sensitivity. One 2024 study identified elevated ankylosing spondylitis risk variants, specific mutations associated with autoimmune spinal disease, in a South Indian endogamous community with a census population of over 1 million people. The variants had no equivalent entry in any global reference database. Their elevated risk had been medically invisible until this decade. The lock that preserved the past is the same lock that delayed the medicine. And there is one more layer to this. The Neanderthal ancestry in Indian genomes, the highest variation of any non-African population on Earth, is not evenly distributed. Some communities carry specific Neanderthal segments that others do not. Segments that, in other populations, have been linked to immune response and inflammatory disease. which means the genetic memory of an interbreeding event 50,000 years ago is shaping disease susceptibility in specific Indian communities in 2025. And the research identifying which communities carry which segments is not finished yet. Yet. That word is doing a lot of work in the science of Indian archaeogenomics right now. UC Berkeley, June 2025. The paper goes out. 2,762 genomes, 50,000 years, 24 million novel variants. The press coverage is substantial. The senior author, Dr. Priya Moorjani, is quoted in Berkeley News. The study fills a critical gap. It reshapes understanding. It will inform precision health strategies. All of that is true. And then the 2,763rd genome is not sequenced. The northeastern tribal communities of India, the populations with the highest concentrations of AASI ancestry on the mainland, the communities that could most precisely reconstruct the ghost lineage that predates everything else in this story, remain outside the dataset. Not through negligence. The consent frameworks, the preservation conditions, the research infrastructure required to include them properly are not yet aligned. It is a solvable problem. It has not been solved. The paper itself says it plainly. India has been underrepresented in whole genome sequencing studies. The 2,762 genomes that represent the largest Indian genomic study in history cover a population of 1.4 billion people. That is one genome for every 507,000 people. The comparable European coverage in global databases is approximately one genome per 30,000 people. The ratio is not a data gap produced by geography or biology. It is the accumulated result of decades of decisions about which human populations a research infrastructure was built to see. Now, before we go any further, I want to ask you something directly. This episode covered 50,000 years of one population's genetic history. Which layer surprised you most? The AASI hunter-gatherers who were here before everything else, the Harappan builders who turned out to be the primary source of ancestry for 1.4 billion people, or the endogamy lock that sealed all of it in place? Tell me in the comments. I read them, and I mean it. Your answer shapes what we build in the next episode. If you have South Asian ancestry, any fraction, any generation, you carry a segment of the AASI lineage that has been in South Asia for 50,000 to 65,000 years. You carry a layer of Iranian farmer ancestry from before farming was invented. You may carry a steppe pastoralist Y chromosome whose original owner rode across Kazakhstan 4,000 years ago, and somewhere in that combination there is a Neanderthal segment, specific to your community, not in any database, not yet named, that has been waiting 50,000 years to be read. The science to read it exists. The infrastructure to apply it to your healthcare does not. Not yet. The one population that would allow scientists to fully reconstruct the AASI ghost lineage, the oldest continuous human ancestry on Earth, 65,000 years, essentially undiluted, has never been fully sequenced. They are alive. Their DNA is intact. The study that would complete the oldest chapter in human genetic history has not been funded. The variable that would resolve the deepest question in South Asian archaeogenomics is not missing from the subcontinent. It is missing from the budget.